Hello, everyone, and good morning. Thank you for joining us for the first AGU Biogeosciences Early Career Spring Seminar. We're very grateful to have y'all here. We're going to have three talks for you today. First, we'll have Eric Levinson. He is a PhD candidate from the University of Oregon, whose research explores the intersections of hydrology, Arctic environmental change, and climate risk. His work leverages remote sensing, deep learning, and statistical modeling to better understand the shifting Arctic systems in a warming world. So please give a warm welcome to Eric Levinson.
All right, thanks so much. I'll share my screen. Okay. All right, well, thanks so much for that introduction. I'd like to start by also just thanking all of you for joining today. I've really been looking forward to this. I'm going to present about interactions between the distribution of permafrost and also lakes and ponds across Alaska, and sort of surprising ways that glacial history can influence that relationship. So I think this image of Summit Lake, it's right along the Richardson Highway with Golconda Glacier in the background. It's kind of a nice primer to begin thinking about this concept that a lake's long-term history can influence its response to modern-day disturbances such as permafrost thaw. I'm also going to dedicate some time to introducing the new Alaska Lake and Pond Occurrence data set, which we recently produced and serves as the foundation for this work. So some important background, some may be familiar, but just the idea that warming permafrost thaw causes lake area change. And this happens through processes tied to changing erosion rates, hydrologic connectivity, as well as vegetation dynamics. And these processes, such as thaw slumping, lateral breaching, vertical drainage, these can cause lakes to appear and grow as well as shrink and disappear. So the expected trajectory is that initial thaw causes lake expansion followed by drainage, and a recent extensive review led by my collaborator Elizabeth Webb found that diminishing lake area is the dominant overall trend that we see, and that's kind of an important fact for the rest of this talk is just that permafrost thaw overall seems to cause decreasing lake area. And so these changes have profound consequences for carbon cycling and ecosystems and climate feedbacks in the Arctic boreal region. So, given these implications, it raises the question of just how widespread thaw-driven lake area changes are across the northern permafrost zone. So, in other words, can we scale our understanding from previously selected and chosen study sites to a regional scale where we see much more physiographic diversity? So that question presents somewhat of a technical challenge because these changes to lakes occur along very complex shorelines. They can be kind of really fine resolution, and they can also be obscured by other factors such as seasonal fluctuations in lake extent. This means that we need to be able to identify and monitor lakes and ponds as discrete components of the landscape with very high detail, but across really large and diverse settings. So that's part of what motivated us to produce the Alaska Lake and Pond Occurrence Dataset, or ALPOD. And basically, I'm going to spend a little bit of time now just sort of talking about its characteristics. So its core product is this lake and pond open water occurrence image. It's derived from Sentinel-2 imagery. And I'm happy to sort of talk about the dataset construction if anyone has questions during the Q&A. But each pixel in the resulting image has 10-meter resolution, and it records the percentage of time that location was open water and attached to a lake or pond from 2016 to 2021. So this gives us some nice detailed information about the geometry and variability of lakes and ponds. And I think that can help us make some nice inferences about important processes like lake formation or drivers of variability. So here's just another local scale example. This is a floodplain of a small tributary to the Yukon. And I just wanted to show kind of what the data set and the vector product looks like. Sometimes that can be more approachable compared to that big occurrence image. This slide is, I think, a little bit of a tangent, but it's just kind of a pet project I've been working on, which is working with the new SWOT data, which is kind of gives us vertical fluctuations to surface water. And I think one interesting application of Alpod is using it as a higher resolution prior lake data set. So in green, that lake in the middle of the green outline, that's the one that's included in the kind of off-the-shelf SWOT product. And then what I did is just process the SWOT data to all these other lakes included in the Alpod data set. And you can see just some statistics about vertical fluctuations there. So an interesting direction for the future. What we're looking at here is just that same data set, 10-meter resolution, but aggregated to watersheds and calculating some statistics across the entire state, just so we can look at some regional scale patterns. The most basic being lake fraction, or density is the number of lakes per 100 square kilometers. And of course, things like shoreline length and size. I think lake area variability, given that occurrence image, is one of the more interesting, aspects to look at. Here, that's shown as average shoreline fluctuation, which is basically a strategy for normalizing for overall lake area. So, given our sort of understanding of permafrost thaw driving lake evolution and this new data set that we have, Alpod, we can ask some basic questions about the relationship between and the spatial interactions between permafrost and lakes and ponds as measured by Alpod. So, given the widely held understanding that permafrost thaw causes lake area to decrease, as well as the understanding that Alaska is all within the historic permafrost zone or last permafrost maximum, which means that we can interpret permafrost extent as the degree of permafrost thaw that has occurred there. We basically expected to see that in areas with more permafrost, we would see higher lake fractions. In areas with less permafrost, we'd see smaller lake fractions. I'll say now that we actually don't find that relationship across the entire state. And that, I guess is somewhat expected but it's an important fact because it sort of lets us know that there are some limits on the controls of permafrost dynamics over lake evolution. So therefore, we investigated whether the permafrost and lake relationship differs based on geomorphic setting and landscape history. And so specifically, we look at glacial history and geologic substrate texture. And that's because of their effects on topography, impermeability, and erodibility of lake boundaries, the things that sort of drive those interactions between permafrost and lake evolution. So today I'm going to focus on this one first being the unglaciated setting with fine substrate. So think sand and silt, things like Alaska coastal plain, Yukon flats, and then sort of compare that to places that were previously glaciated and have more rocky and in some cases bedrock substrate. So starting with that unglaciated fine substrate setting, the takeaway here is that we basically see the patterns you would expect to see given the understanding that permafrost thaw causes decreasing lake area. So if we start on the right side of this box plot and continuous permafrost and go from right to left, you're going along a gradient of increasing permafrost thaw. And in this case, we see a corresponding decrease in lake fraction. Right, so that's sort of the expected thing that more permafrost thaw causes lower lake fraction in this setting. These are just kind of the same results, but this is showing actual example watersheds within the study, the Alpod Lakes shown in blue. And on the left, we have that continuous permafrost. And I think what that's doing is essentially serving to preserve these thermokarst lakes that were formed during historic thaw cycles. And on the right, you can see discontinuous permafrost, and we can start to sort of imagine some of the processes that drive lake area change and decreasing lake area in this setting, such as increased incision on the landscape, lateral breaching of lakes, so lakes now connecting to the river network and then draining out the side. And there could be the possibility of increasing groundwater flow as well, so some vertical drainage. So now looking at this post-glacial setting, the moral of the story here is that we actually just see the opposite relationship. So going right to left, we're going increasing permafrost thaw, and we see the opposite, increasing lake area. So this is somewhat surprising to us. And so now kind of again looking at some example watersheds, this is what we see here is, the possibility that these areas could just be more resistant to the processes of permafrost thaw. One kind of potential hypothesis is that the presence of permafrost actually delays the onset of the landscape evolution processes that normally follow glacial retreat, such as the formation of kettle lakes. And then once the permafrost thaws, then those processes can take place and more lakes will start to form. Very similar to just thermokarst lakes. So this is just to kind of highlight the contrasting relationship between permafrost and lake fraction in these different settings. Now, one sort of weakness of this study, of course, is that thus far it's been kind of a purely spatial analysis. And of course, this is a temporal process. So basically what we did is we decided to look at previously published studies of lake area change in the permafrost zone. And what we found is that, first of all, the vast majority of studies took place in this unglaciated setting. And this is despite the fact that the majority of the Arctic, even excluding Greenland, was previously glaciated. Think like all of Canada. It's a big landmass. And then furthermore, the overwhelming trend of lake area declines, which has become sort of the dominant storyline, those were almost all found in this unglaciated setting as well. So this basically indicates that permafrost controls over lake area may be sort of confined to these settings, or at least in that expected relationship. This sort of story of disappearing Arctic lakes may be not reflective of the entire Arctic and maybe kind of just focused on some specific geomorphic settings. So I'll leave it there in case there are any questions. Yeah, I really appreciate y'all joining.
Thank you so much, Eric.
Yeah.
That was great. Yeah, so I'll open the floor up to questions. What questions do people have for Eric?
Hi. Oh, Ariel has.
No, go ahead, Kaylin, and then we can follow up with Ariel's.
Okay. So my background is not in kind of like this area, but I'm curious about the methodology that you use to create this Alaska land and pond occurrence data set.
Mhm.
Is it like really only of interest in an area where you have lots of permafrost, or could it be applied to other areas where you might have like drought affecting like the size and extent of lakes and ponds?
No, in terms of, I'll just go. This is kind of like a method slide, but in terms of just like producing the actual data set itself, that isn't specific at all to sort of a permafrost setting. It could be, I think, applied. It would be a question of like, for example, in urban areas, some of like the water classification algorithms, like whether or not they perform exactly as well or not. In some cases, like the contrast between vegetation and water is useful. But that being said, the workflow itself isn't really specific to a given region, and I think it could easily be applied. Yeah.
Cool, thank you.
Yeah, totally.
And then I think we have Ariel's question in the chat, and I can just read it out for you,
Oh,
Eric.
sure. Yeah, I didn't see that.
Yeah, so how do you disentangle the effects of permafrost extent from other factors like topography or precipitation when analyzing lake and pond distribution?
Yeah, it's a good question. So basically what we did for the especially the interactions with topography are really difficult. So basically what we did is we looked at these relationships at both only low elevation subsets, so like under 300 meters, and excluding high relief settings, so all the mountainous areas, because there is the possibility that especially that pattern we saw in the post-glacial settings could just be sort of like an elevation signature. We found basically that the relationship held at just the lower elevation subset as well. And Elizabeth, my co-author, also kind of she's expanded this to look at more kind of the Pan-Arctic scale and is finding similar things as well. I think the precipitation is more of a challenge for the studies that are doing kind of like the temporal studies because they're looking at kind of specific timestamps and kind of trying to like evaluate trends and disentangle what's driving the trends in lake area change, whereas this sort of spatial thing is more about kind of like the long-term evolution of the distribution of lakes. And I think one nice thing about the occurrence raster is that it kind of gives you a snapshot of like how variable those lakes are as well. And of course, that you kind of need to choose a threshold to do kind of a comparison across regions, which can be challenging in its own right. It's a good question.
Other questions? If not, I think we have a couple more minutes. I'm curious about, so when you're speaking about this kind of bucket of unglaciated and post-glaciated, I know in Alaska and in Siberia, a lot of the permafrost extent is dominated with like this Yedema
Mhm.
permafrost. And I'm curious, so that has like really high ice and carbon content, and I'm
Totally.
curious like um it and that was unglaciated, and I'm
Right.
curious, like, were there any distinctions between the Yedema landscape and non-Yedema other unglaciated landscape, or was Yedema like a factor, or did you
So
disentangle
Yedema, I
those?
think basically is captured by this like unglaciated fine substrate setting that I
Yeah.
described. I think we basically there's like infinite ways that you can like divvy up the landscape and try and
Hmm.
classify. And just based on sort of like the data that we had available across Alaska, these definitions became I think that sort of the easiest to work with. But I
Totally.
think that that's a good point that the Yedema landscapes and Yedema permafrost really is, I think, included in that sort of unglaciated, mostly fine substrate setting. And there is a reason why most of the studies take place in that type of setting because that's where we see the trends that sort of like are really interesting, such as draining lakes and that type of stuff.
Yeah.
Yeah.
Hmm.
And
Very cool.
also the carbon content is I think a really valid reason to sort of decide if you're going to choose a specific study site, it makes a lot of sense to choose places with high carbon content.
Yeah, absolutely. Very cool. Well, I think if there are no more burning questions, we can wait till the end, and we can move on to the next speaker. Take it back to you, Ariel.
All right.
Thank you
Thanks.
so much, Eric. Thanks. Next, we have Alice Pupin, who's a postdoctoral researcher at the University of Padova. She's working on a project on the resilience of coastal ecosystems under climate change. Her research focuses on geomorphological dynamics and carbon storage in tidal ecosystems. So thank you, Alice.
Thank you. I'm going to share the screen. Okay. Can you see it? Okay. Good morning, everyone. And of course, thank you for the opportunity to speak today. I'm pleased to share some insight from our research on blue carbon dynamics in the tidal marshes of the Venice Lagoon. I'll begin with a brief overview of what blue carbon is and why it matters. Then I'll introduce the work my research group has been doing on organic carbon in tidal marsh soils. So let's start with what blue carbon is. Blue carbon refers to the organic carbon stored in marine and coastal ecosystems, which have a crucial role in the global carbon cycle. Coastal blue carbon ecosystems like mangroves, seagrasses, and tidal marshes have been increasingly recognized for their ability to sequester and store carbon, helping to mitigate climate change. At the same time, they provide a wide range of environmental, social, and economic benefits that support many coastal communities. However, these ecosystems are also under threat from both climate change and human activities. For this reason, Strategies to conserve and restore blue carbon ecosystems are gaining momentum as natural solutions to fight climate change. These strategies can help avoid CO2 emissions, enhance carbon capture, and provide, as we said, a range of co-benefits from biodiversity conservation to coastal protection. As interest in carbon markets grows, blue carbon projects are also becoming valuable funding opportunities for conservation and restoration. But to implement these projects effectively, we need to improve our understanding of blue carbon, especially how it varies in space and what factors drive its accumulation. Toward this goal, my research focused on the analysis of organic matter and carbon content in tidal marsh soils. Tidal marshes are intertidal coastal ecosystems developing worldwide along low energy temperate coastlines. They are characterized by sedimentary platforms covered by herbaceous vegetation, regularly flooded and drained by tides. The high potential for carbon storage and accumulation in marsh environments is primarily driven by high biomass productivity and vertical accretion, with up to 90% of stored carbon found in soils. Carbon captured through plant photosynthesis is buried and preserved in soils for long timescales, with sea level rise providing continual vertical accommodation space for additional carbon storage and saturated soil leading to slow decomposition. Our study site is the Venice Lagoon in the northwestern Adriatic Sea in Italy. This is the largest lagoon in the Mediterranean and is a shallow microtidal basin with marshes, tidal flats, and channels, hosting the iconic city of Venice. The lagoon's current landscape is the result of both natural processes and centuries of human intervention, including river diversion and canal construction that have significantly altered sediment dynamics and contributed to marsh loss. In our study, we analyzed around 1,500 soil samples for sediment cores collected in different areas of the lagoon to capture variability at both marsh and lagoon scale. To assess blue carbon, we used two key indicators, carbon stock and carbon accumulation rate. Carbon stock refers to the total amount of carbon stored in a system, in our case, marsh soils, at a given point in time, and it's typically measured in 1 meter topsoil. Carbon accumulation rate, on the other hand, tells us how much carbon is being added and stored over time and help us understand the potential of an ecosystem in sequestering carbon. As for carbon stock, we found that tidal marsh soils in the Venice Lagoon store about 171 megagrams of carbon per hectare in the top 1 meter layer, a value which is similar to carbon stocks found in temperate forests. However, we observed a high degree of variability. This variability is mainly driven by differences in organic matter content, which depend on current environmental conditions, of course, but also on the development history of each marsh. In fact, this history is reflected in the vertical distribution of organic matter in the sediment core, and we can reconstruct it through sedimentological analysis. Let me give you some examples. In areas dominated by halophyte herbaceous vegetation, we found lower organic matter levels than in zones dominated by reeds, which grow where freshwater inputs reduces salinities. In marshes with lower accretion rates, we found thinner marsh layers, often transitioning in older tidal flat sediments below, with organic carbon content rapidly decreasing. In contrast, marshes with higher accretion rates had thicker thicker marsh deposits and more irregular organic vertical profiles. In some locations, we even observed buried remains of high carbon content reed vegetation from when rivers still flowed into the lagoon prior to diversion. Our findings show that different tidal environments have different blue carbon potentials, with marsh deposits having significantly higher organic matter than tidal flats and transition zones. This highlighted measuring carbon stocks down to 1 meter in tidal marshes must be done with caution, as variation in depositional environments over time in dynamic tidal systems can strongly influence carbon content. But variability remains high even within marsh environment and is controlled by multiple factors. From our observation, we proposed a general framework to explain organic matter variability in marsh soils. The main driver is, of course, the input of organic material, vegetation, but inorganic sediment inputs also play a crucial role in marsh formation. Relative sea level rise, water level fluctuation, and sediment availability all affect both organic and inorganic accumulation. And these processes influence marsh surface accretion, and in turn, plant growth and organic matter inputs. Then hydrodynamics, including tidal flows and river discharges, shape vegetation patterns and influence the inputs of autochthonous and alloctonous organic material. These aspects, together with sediment properties like grain size, also impact organic matter decomposition and so its conservation in the soil. All of these factors vary across both space and time, from lagoon-wide scales to individual marsh scale. Now, moving on to the second assessment indicator, carbon accumulation, we attempted to estimate how carbon is being added and stored in marsh soils over a medium term. And to do this, we calculated the product of soil carbon density and the accretion rate of the reference sediment layers. Our results suggest that the tidal marshes of the Venice Lagoon, 43 square kilometer, as a whole, may sequester over 13,000 tons of carbon per year, which is equivalent to roughly 20% of the local annual CO2 emissions from the Venice airport. Zooming in at the marsh scale, one of our recent studies found also that abandoned channels within tidal channel networks cutting through marshes can act as hotspot for carbon accumulation. Once these channels become inactive due to natural network dynamics, they tend to fill quickly with sediment and organic material. And this leads to carbon accumulation rates more than 5 times higher than those in the surrounding marsh areas. So also channel networks may have an important role in carbon dynamics at local scale. To conclude, our research confirms that tidal marshes have a high blue carbon potential, but also that this potential varies significantly across space, shaped by numerous and interconnected drivers. Understanding these patterns is essential to identify carbon hotspot, anticipate potential carbon losses from marsh degradation, and design effective conservation and restoration strategies. And these can support better decision-making for coastal management to the benefit of both people and natural environment. This was a brief overview of some key results from my research. I thank you for your attention, and I'd be happy to take any question. And feel free to reach out to me. This is my address if you'd like to discuss further. And here also the QR code to the website of our research group, where you can find other information. Thank you.
Thank you, Alice. That was great. All right, we'll open the floor for a couple questions. Yeah, Keelan.
Hi, Alice, great talk. Thanks for sharing. I was just wondering if I was noticing in all of your very cool graphics where you're talking about carbon accumulation, you see like there's this vertical, like you can physically see it vertically occurring where I'm assuming like the level of the salt marsh area would be also getting higher. I'm just wondering if like there's any talk of like having to weigh the benefits of carbon accumulation from like a carbon sequestration standpoint, and then like what are the effects of having like a vertically higher salt marsh, if there's like any sort of tension there, or if it's like both considered good things.
Well, it's good question because vertical accretion in salt marsh is a natural process that allow them to keep the pace with sea level rise, and for recent history told us that salt marshes were able to keep the pace of sea level rise and accrete and keep their equilibrium with sea level rise. This means that they remain in that at that elevation that allows them to receive sediment inputs during high tides, but they, of course, with these sediments, they are able to accrete vertically and not to drown due to sea level rise. And this is important also for carbon accumulation because, as I said, these vertical accretion gives the opportunity to have, let's say, space to accumulate more carbon and not to saturate the soil with the carbon. But as I said, it's a matter of equilibrium. So if marshes grow too much, they could become something else that is not anymore a salt marsh because the tide it's not anymore able to flood them. But on the other hand, if they are not able to grow enough to keep the pace with sea level rise, they would drown and they will disappear, basically.
Okay. Gotcha. Okay, cool. Thank you.
Thank you for the question.
Other questions?
I have one.
Yeah, go for it, Sophie.
Can you actually hear me? Yeah, okay. Sorry. Issues with my audio. Anyway, thanks for the talk, Alice. I was wondering, did you see any anthropogenic imprints in your samples due to the proximity to Venice, or was that something you haven't studied, or it was not visible in the data?
Yeah, it's a good question because, of course, the Venice Lagoon is highly impacted by anthropogenic pressure, and also this pressure has a long history. And as I briefly said, the lagoon was modified with, for example, river diversions, like in these areas, and also canal construction and jetties construction, and these alter the hydrodynamic and the sediment distribution inside the lagoon. And as you can see in this graph, salt marsh area decreased a lot in the last couple of centuries because of these interventions. And these it's also visible in organic matter distribution because, for example, we observed, for example, here in the Southern Lagoon, we observed that previous to river diversion, there was a brackish environment with reeds growing and giving a higher input of organic matter. And then when rivers were diverted, the vegetation changed and organic matter content was reduced. And nowadays, another important impact that we observed in carbon sequestration in salt marshes is the impact of storm surge barriers that were constructed in the Venice Lagoon to protect the city of Venice from flooding. And they are they are 3 barriers at the 3 inlets, which disconnect the lagoon from the sea during storm surges, when the level of the water is too high. And doing these, they also prevent marshes from being flooded during storm surges. And some colleagues observed that these storm surges are the moment when marshes receive the higher amount of sediment. So storm surges are the most important contributor to vertical accretion. And if we reduce vertical accretion, we also reduce the capability of marshes to accumulate carbon. So yeah, of course, human impact have an important affect importantly these organic carbon dynamics in marshes, and we need to consider all the all the impacts of the coastal management actions to have sustainable solutions.
Thank you.
Thank you.
Great, thank you, Alice. That was great. I think we should move on to our last speaker, so I'll take it back to Ariel.
Thank you.
Thank you, Alice. That was an amazing talk. Next up, we have Qipei Shangguan. He is a postdoctoral scholar at the Woods Hole Oceanographic Institution, specializing in the development and application of autonomous chemical sensors for marine environments. His research focuses on marine metabolism, air-sea gas exchange, and the advancement of time series data processing, techniques to better understand biogeochemical processes in aquatic systems. So thank you again, Qipei, for participating in our seminar.
So I'll start share my screen. Okay, let me disable. Okay, thanks for having me. Yeah, I'm from a marine institution, but I'm going to talk about the river research, which I did during my PhD, the freshwater river carbon cycling. I'm going to talk about an important process called metabolism, and how we are going to estimate the metabolism. And why we care about rivers, rivers are very important ecosystems, and they connect the terrestrial environments and also the coastal oceans. So a long time ago, people thought that rivers are working like pipelines. They are receiving ground inputs from terrestrial environments such that they have lots of carbon, and then they directly deliver the carbon to the coastal oceans. So they like conductors, like pipes to transport the carbon. But later on, and more studies found that rivers and streams, they emit a lot of carbon dioxide into the atmosphere. And this quantity is actually very large. Oh, sorry. And I'm trying to show my... And this quantity is actually very large. They emit a lot of carbon dioxide into the atmosphere. And they also form the precipitates, the calcium carbonate, into the sediment for storage. Meanwhile, during the transport from land to the oceans, there are lots of biological activities, photosynthesis and respiration. Photosynthesis and respiration, they change the carbon forms that are organic carbon and inorganic carbon. They respire organic carbon, produce CO2, and the CO2 get evaded into the atmosphere. So it's like a reactor. It's not just a pipeline. The river is actually much more complicated than we thought. So I'm going to focus about this process called metabolism during the photosynthesis and the respiration, the conversion between CO2 and the organic carbon. And the oxygen is one of the products or reactants for the metabolism. So during this process, oxygen has strong Diel cycles driven by the lights. During the daytime, the photosynthesis generates oxygen, the oxygen levels increase, and the respiration, the decrease, consumes the oxygen concentrations. So nowadays, there are lots of oxygen data in rivers, and the models used to estimate metabolism from oxygen are very robust. Because the oxygen data variability, we can calculate the rate of photosynthesis and the respiration. Here comes a question. If we know the oxygen variability and the oxygen-based metabolism, how well we can predict the CO2 variability? So this is a very simple equation, and it is not well balanced because it doesn't have the stoichiometry. The stoichiometry we are using is called PQ and RQ. It's called photosynthetic quotients and respiration quotients. That's the ratio between oxygen and CO2 changes. And people normally think in the rivers, PQ and RQ equals to 1, but PQ and RQ are actually more complicated. It has a large spatial temporal variabilities. And how do we actually quantify the PQ and the RQ? How do we use the oxygen time series and estimate metabolism and convert to the CO2 variabilities? So this is a question, and what's the uncertainty from this conversion from oxygen to CO2? Because we don't know the PQ and RQ. The method we developed is called a Lagrangian or two-station approach for rivers using autonomous sensors. I say this is a rivers. We have an upstream sensor monitoring station, and we have a downstream sensor monitoring station. This is called two-station because there are two monitoring stations. And for every water parcel, the rivers flows from upstream sensor site to the downstream sensor site. It takes the length of the travel time, tau. And for each sensor site, we have sensor measurements of oxygen and CO2, high frequency measurements of CO2 and oxygen together. And if we know the sensor, if we know the what's the parcel travel time and know the oxygen difference over the reach, we can calculate how the chemical changes like oxygen and the CO2 over the reach length. And we use the DIC here to quantify the total inorganic carbon changes from CO2 and the alkalinity. And we can build the models to solve metabolism both from oxygen and from DIC. I will show you an example later. This is the, we conducted our research in the Clark Fork River in Montana. This is a map of the Clark Fork River, and we select 3 different reaches called Milltown, James, and Garrison in the Montana. So for each of the reach, we require at least 2 monitoring sites because we want to build the 2 stations. Sometimes we have 3 different sites, so we can pick any of the 2 to form a 2-station model. And we select the 3 different reaches to represent the different complexities of hydrology. For example, the Garrison, there is a wastewater treatment plant. And in the Milltown Reach, we have a dam above the monitoring station, so we can test the model efficiency, how well we can estimate the metabolism from the sensor data and compare the metabolism results among 3 different reaches. This is an example of the sensor data. We have very high quality, high frequency monitoring of PCO2 and dissolved oxygen, and also a whole bunch of other variables like temperature part to drive the models. And we calculate, this is 1 day of the data. We can calculate the difference between upstream and downstream for dissolved oxygen and PCO2. Then we build a model to fit the data. For DO, we can calculate gross primary production, ecosystem respiration, and the net ecosystem production, the difference between the 2. And we can do the same thing based on the carbon. So later on, we can compare the metabolism results from DO and the DIC to quantify the PQ and RQ. And here we show PQ and RQ not equal to 1. So most studies assume PQ and RQ equal to 1 and directly just use oxygen to predict the CO2 dynamics. But if we model them together, we show they are not equal to 1. This is, for example, like the comparison of GPP-DO and GPP-DIC for all 3 reaches. The PQ actually above 1, sometimes it's close to 2. We can do a similar thing for ecosystem respiration. And the slope, the inverse of the slope represents the respiration quotient, RQ. They are less than 1. They are not equal to 1, as most people assume. And because of the PQ and the RQ difference, different from 1, we have net ecosystem production predicted from DO and net ecosystem production from DIC. They are not equivalent because PQ/RQ are not equal to 1. If net ecosystem production is negative, that means it's producing CO2. Producing CO2 on a daily basis and increase the CO2 emission. That's true for all three reaches. And the important point is that the NEP-DO, the red color over here and the green color over here, NEP-DO and NEP-DIC, they are not equivalent. That means if we assess the contribution of metabolism to the CO2 emission, and if we use DO and the DIC, they have different conclusions. And when AEP equals to 0, that means there is a net balance of carbon and inorganic carbon from biological activities. We assume this amount of carbon actually comes from groundwater inputs. So if AEP is a negative, it adds more CO2 from the organic carbon. So the important point is that the different metabolism results due to the PQ and the RQ, they provide different conclusions into the contribution of metabolism into the real carbon cycling. Let's think more about this. Why PQ and RQ not equal to 1? So first of all, it depends on the organic metabolism. It can be a sugar, very simple sugar, such that oxygen, carbon, the ratio close to 1, but it can be other organic components like proteins and the lipids. The carbon-oxygen ratio actually varies. So another important point is what we are measuring in PQ and RQ actually is on an ecosystem level because there are lots of other processes happening at the same time with the aerobic metabolism, like denitrification, for example. It produces CO2 and it occurs on the low oxygen. Like calcite formation, it is just a change CO2, but has nothing to do with the oxygen. And these lots of processes in the collection, they will drive the oxygen and CO2 variabilities, like this picture shows. So if we directly measure oxygen, it's difficult to predict the CO2 changes because there are so many so many processes that are happening together. So the better strategy is to measure oxygen and CO2 together and model the metabolism. So we should distinguish the PQ and RQ concepts from the substrate level to the ecosystem level. Another point I would like to mention is that the respiration, because most of the models right now, they assume respiration is a constant through 24 hours. But there are studies suggesting that respiration could change over one day. This is a dial change of ecosystem respiration using the isotopic methods. And most of the models, they assume respiration are similar between daytime and nighttime. So we estimate metabolism, it can cause some errors. So, and if we don't know respiration very well, then it's difficult to quantify photosynthesis because most of the time, photosynthesis and respiration, they occur together. And if we know ER has some errors, then JPP must have some errors. So to summarize my today's talk, the first day I talk about the real-world functioning of processing carbon. It's not just a pipeline, transport carbon. Because of the ecosystem metabolism is transforming the organic carbon to inorganic carbon by respiration and the CO2 emitted to the atmosphere. And how do we estimate this process? And most studies using DO directly, and it requires a secondary stoichiometric conversion using PQ and RQ. And they assume PQ and RQ equals to 1. And we develop models to estimate metabolism from PCO2 directly. And we compare the results from PCO2 and DO and to quantify PQ and RQ. They are not equal to 1. So we should be cautious of this. So finally, from a scientific level, we should think about the PQ and the RQ. They are different when we're talking about on the ecosystem level and the biochemical level. On the ecosystem level, there are multiple processes occurring, driving oxygen and CO2 variations. So they are not necessarily equal 1. They are more complicated than we thought. So a good strategy is to measure PCO2 and oxygen together instead of just measuring oxygen. Today is my talk. Thank you.
Thank you so much, Chipre. was a great
The-
talk.
Mm.
Let's see. All right, well, we have some time for questions.
I have a question.
Go for it, Ariel.
So you were talking about the uncertainty in getting your measurements. Are there any emerging technologies that are coming out to help provide more certainty in those measurements?
I
Specifically,
think there are.
dissolved oxygen cycles.
I think oxygen sensors are widely available right now, and the availability of CO2 sensors are also increasing. But increasing, but it's still not that available as oxygen. And I think another issue for CO2 measurements in the rivers is potentially due to the duration of the deployment. But as the time goes by, the sensor gets baffled. The measurement quality is decreasing. And actually, sensors are measuring well in the lab. As time goes by, and more scientists develop these CO2 sensors. The problem is that how well we can deploy in the rivers, more complicated, more dirty environments.
Sophie, are there any questions in the chat?
Yes, there's one in the chat.
Oh, cool.
Read it out. It's from Roberta. Great presentation, thanks. Do you incorporate losses to the atmosphere into your metabolism models? And can the difference between DO and DIC be related to different water air gas exchange rates, like losses between your two measurement stations?
Yes, this is a great question. We do incorporate the difference gas exchange rates. Like for the two-station model, we have an upstream sensor, downstream sensor, and their signal difference between the upstream and downstream equals to metabolism and gas exchange. So we do incorporate gas exchange into the models, and they are different between DO and the DIC. So, in other words, we should get rid of gas exchange effects to assess the effects of metabolism.
And then there's a short method question, like at what depths do you take your measurements?
What depths you do measurements? Normally, our river is around 0.5 meter to 1 meters, and we put our sensors in the on the bed of the river, just the bottom of the river. But the river depth is around 0.5 meters to 1 meters.
Awesome. Well, I thank you so much, Chipe, and to all of our speakers. I know we just have a few more minutes, and I know that there was some interest in just having a brief discussion. So I thought I would just tee it off and allow each of the speakers to give maybe a response to kind of an overarching kind of big picture question. Obviously very brief because we only have a couple of minutes. But we've heard some great talks in the water world about permafrost-affected lake dynamics in the Arctic, mediated by glacial history and impacted by permafrost thaw. We heard about coastal tidal marshes in Italy and their importance to sequester carbon and the factors that drive this variability and vulnerability to carbon sequestration. And finally, we heard about river systems in the Western US and understanding how rivers process carbon, not just transport it, and understanding how to better predict this metabolism process. So really great range of questions in the water world. So my big picture question is, what are the most pressing knowledge gaps in the water world in relation to threats to aquatic ecosystems under the changing climate? So I know it's a really big picture question. So if one of our speakers want to just give kind of their 2 cents.
Can I start first? Oh, yeah, okay. I'll briefly talk about this. Actually, I was doing the river research when I was a PhD, and doing the coastal research right now. And for the climate change, especially in the dynamic ecosystem, I mean dynamic like rivers, and the coastal shallow estuaries, like the salt marsh tidal waters. I think it's different. The assessing their climate signal are different from the open ocean. And I think the difficulty for assessing the climate signals are the availability of the data and the duration of the data. And like for the sensitive performance, if you just collect data for several tidal cycles, you won't be able to see the climate signals. And we don't have a very continuous long-term observations sometimes for our study site. This is true for the coastal ecosystems and the rivers, but in the open ocean, they have a different story. They have a long-term monitoring station. It may be easier to study the climate signals.
Great.
Mhm.
Eric and Alice, any thoughts?
Yeah, sure. So it's a good question. I think in terms of, I think for me the question of data availability and sort of long-term records is always an outstanding question. I've felt like for a long time as sort of like a satellite-based kind of empirically focused researcher in the world of hydrology. My focus a lot of times has been kind of more on like water quantity and sort of water storage and that type of focus, with more kind of like a hydrology background. And in that world, I think, there's this limited understanding you can get from the gauge data that we have, which is part of the reason I went to sort of pursue this satellite-based research. And then there's a really limited kind of understanding you can get from like a two-dimensional approach in terms of like what I presented today in terms of using lake areas, for example, or river surface area as another example. But I think moving forward, one of the biggest knowledge gaps that we have the potential to help close is to move towards, at least for hydrology or remote sensing or surface water remote sensing, I think the next frontier is to start talking in terms of like actual volumes, and trying to like be able to get good estimates of river discharge and lake and reservoir storage. I think with the new SWAT satellite, that's kind of a really important direction that we need to be able to go and get our capabilities sort of at the point where they're trustworthy for looking at individual water bodies. And then people like everyone here who have more of the sort of expertise in the biogeosciences can sort of like, if that's a way that we can integrate data to start to better understand carbon cycling and scale from our like sort of specific point-based measurements to a regional and global scale, I think that's kind of a great direction for us to go.
Excellent. Alice?
Yeah. Thank you for the question, which is quite wide, but I agree with what with what previous speaker said, and I think that both data availability and integration of different scales are really important points, because also as concern organic carbon in coastal environments, there are some models using, of course, also remote sensing data that try to predict organic carbon sequestration and storage at global scale. But I think we lack data from some regions, and so the result of the model could be not really appropriate for some region that can be different from others. For example, I think the like Mediterranean or the high latitude regions. So I think that remote sensing data can help a lot in this sense, but we need to increase, I think, in some areas the link between remote sensing data and field data to fill the gap.
Thanks so much. Ariel, I don't know if you have any concluding words. I know we're over time. Oh, Ariel, you're muted. You're muted.
Thank you for that. I hope you guys can see my shared screen. So I wanted to share this QR code with everybody real quick before we ended. And it's just giving you a link to all of our social media and contacts for the AGU Early Career Committee for the Biogeosciences section. I really want to thank our speakers today, Chipe, Alice, and Eric, for giving us our inaugural talks for our spring seminar. Like, we really appreciate y'all participating and giving these talks. But if you'd like to be featured as a scientist on any of our social media pages, or if you would like us to feature any of your manuscripts or articles you're writing, please scan the QR code and reach out to us. We're really here trying to build community for our fellow early career folks, and we're trying to build this community, make it bigger and stronger. I hope you guys all have a lovely Wednesday and enjoy the rest of your spring. Bye.
Thank
Thank you,
you.
everyone.